In this series of conversations, we'll be discussing food sustainability with guests who bring a deep understanding of the environmental and cultural challenges facing our society and creative ideas on how to address them. I'm Ash Sweeting and welcome to the Ash Cloud. Today we are joined by Todd Callaway, a rumen microbiologist from the University of Georgia. Todd has spent his career trying to better understand the complex, competitive and collaborative microbial environment in the digestive tracts of grazing animals. His work highlights the huge impact these billions of microbes have on the food we eat, the environment, our health and the climate.
Ultimately, like for forage degradation, methane production helps speed that process up. So methane production is actually beneficial to forage degradation. They're really inextricably intertwined.
Do we know what we're aiming for?
Because everybody wants to come to us and say, what's a good microbiome? Like, what's a good microbial population? And there's not a simple answer for that. That is a really complicated question because that's going to vary a lot depending on what's your lens that you're using. And I teach them, from a microbial perspective, it's one thing, from the animal, but the human perspective is another. And sometimes those align, sometimes they don't.
How much do we actually really know?
But we still are finding there's more that we don't know than what we do. There's not a time I don't look in the rumen and I'm not just dumbfounded by something that we see.
How is the microbiome set?
There is an interaction between the animal's genetics and what host microbial population they have. We do not understand that, but it looks like about 60% of the microbiome population, of that microbial population, is encoded for by host genetics. So there's some really profound communication of some form between the microbe and the host.
Where's the opportunity?
That my argument is that core microbiome, is it really important because it's the same, it's present in all species? Or do we want to look at the ones that vary? Is that your difference?
What does the future look like?
So can we come up and look at an animal, pull it up in the chute, do a fecal sample and or a ruminal sample and say, this has got a good microbiome, we don't need to do anything. This one's got a bad one, let's give them product X. This one's got a good one in the rumen, it's got a bad one in the feces, let's give them product Y. And, that I think that's one of the places where it's almost tailored medicine for these animals. But that's going to require an investment in time, contact, working with the animals.
Collaboration is key.
That's one of the great things of the world of animal science and ruminant nutrition especially, is it's such a small world and it's a family. And everybody kind of talks to each other and listens. And, I could call some of my competitors right now and they'd send me samples and they'd help me out and vice versa.
Young people are the future.
Millennials and Gen Z want to solve problems. That's their goal. They're kind of, for want of a better term, and it's always dangerous to say things about a generation, but they're kind of crusaders. They want to make the world a better place. And at this stage where they've got the enthusiasm and the energy and the belief they can do it, and there's really no greater force in the world.
Todd, thank you very much for joining me today.
Sure, it's my pleasure.
When I heard you speak at Cornell at the Dairy Conference in October last year, you said that about 10 or 15 years ago, the microbiome, the animal microbiome, the rumen microbiome used to be a big black box. And now it's just a very, very, very dark box. So what's changed that? What do we know? What do we need to know? What are the opportunities? And if you just share with us your thinking on where our understanding of the microbiome, what the potential of understanding it better is, and what we need to do to get to that stage.
Okay, well, that just covered about 20 years worth of research in one question. Really, the rumen was always a black box in the sense that we put something in, we get something out. And research started in the 1950s and 60s trying to understand what was going on, where we would throw something in and we'd see certain end products. So that allowed us to see that interaction between the microbes and the animal, and that fermentation, how it makes changes in feed stuff that affects the animal. And we did this, our predecessors in the rumen world did this for years. And on the human side, or pigs or chickens, easy for me to say, that on that side of the house, they didn't really look at that because they didn't consider it. It was, you feed these animals, they grow, that's good enough. Humans, we feed them, they grow, it's not good enough, or it is good enough. So then we would take that information of what's this microbial ecosystem doing, and it's a really complex, very dynamic ecosystem in the rumen, and we used the best tools that we had of look at different populations by plating and dilution, all the old-fashioned techniques, and we began to understand the rumen pretty well. And that's the world I came up in, was doing the microbiology, that old-fashioned micro. And then we started having molecular biology revolution. And I've stayed kind of a dinosaur in some ways to try to do the growth and the understanding of what the pathways are, doing the biochemistry of it. Because there's a lot of people who come in and they just go from field to field doing molecular biology in one set of animals or one set of proteins. So we started having people looking at specific genes or markers for specific microorganisms. So we started to be able to see things that we couldn't grow. And what we realized pretty quickly is we could only grow maybe 10% of the bacteria in the gut of the cow. And we understood that environment pretty well. And, we were getting that 10%, maybe 15 or even 20% in some cases. So that was the best we could do. And the molecular biologists started finding that, yeah, this looks like that's about the number, 10%. Looks like there's a lot more there that you can't or don't grow than what you do, which really made everyone in the field just kind of throw our hands up and go, well, crap, what do we do with this? And about that point, a new technique came down. It was called pyrosequencing. It was the first of the large scale sequencing, where you could take a complex population, throw it into a machine, and it could figure out what microbes were there. And you were getting a snapshot of what organisms were present. And we were never able to do that before. So suddenly we had this great hammer, and the whole world looked like nails. So for us in the rumen, we're like, this is a natural extension of what we've done. So I started playing with it, and a couple of other folks did too. But on the human side, and looking at mouse models and pigs and chickens, all of a sudden everybody said, oh my God, the microbial population makes a difference in the animal productivity. It can affect carcass quality. It can affect how much animal you get. We found this new revelatory idea, and collectively the rumen nutrition world said, well, duh. We know this, that we've known this for 60 years, guys. Come on, this isn't novel. But all the human people were running around for a while of explaining the novelty of this. And, again, for us, this wasn't novel. This is what we grew up doing. So, there's been a little snottiness on that especially out of our end of, we've known this, but the fact that on the human side, they've got so many more resources that suddenly they advance so rapidly with these tools, where now we've got Illumina, PacBio, and there's basically every 2 or 3 years, there's a new technique coming down. When I first started it, the first time we did anything in the rumen on pyrosequencing, and it was about $28,000 to do, I think it was 24 samples, which was incredibly expensive. I just had a student do something like 250 samples for, I want to say it was $2,000. I mean, just incredibly cheaper now to be able to understand this, and you get more data. It's a lot higher resolution. It's like taking a either your iPhone camera or using the Hubble Space Telescope or now the James Webb Telescope, you can get a lot better resolution on that higher end. And so we're now using those James Webb telescopes to look inwards into these populations, and it's cheaper than it's ever been. So now we almost are reaching that point of paralysis by analysis, that we have so much data and we're seeing there's so much individuality from animal to animal that it's hard to say here's a normal population, because everybody wants to come to us and say what's a good microbiome, like what's a good microbial population. And there's not a simple answer for that. That is a really complicated question. So all that's just a really long way to say we've started shining a light in there by having this amazing new set of techniques that we never could have dreamed before. If the really great scientists like Bob Hungate and Marvin Bryant from the 50s and 60s had these tools, they'd have solved the problem, a generation plus ago. But, the lesser of us that are around now, like myself, we have to make do with what our capacities are mentally instead of being the giants. So we try to move things forward. And it's really nice to be able to see one of the advantages now, instead of the rumen working in or rumen environment microbiology working in isolation, we can look over and see what the pigs are doing, what the chickens, what the humans, the mice, random animals, and we start to be able to see these patterns. And now, with the advent of AI and these large data-driven analyses and cluster analysis, people are now able to go in and take data sets from different species and start to try to see patterns. But then they're coming back to those of us on the ruminant side again. And just a couple of years ago, there was a conference where a lot of the human people were telling me, well, we see this relationship between cellulose and hemicellulose and volatile fatty acids, and that affects animal productivity. You guys never imagined that. And again, the response from everybody in the rumen world is just a collective meh. Welcome to the 1970s, guys. So, there's a lot of reinvention of the wheel, and we did a lot of that pioneering work. So now we're able to talk to these other species people, and they, it's helping them advance very, very rapidly. So, we've been that kind of collective knowledge, but we still are finding there's more that we don't know than what we do. There's not a time I don't look in the rumen and I'm not just dumbfounded by something that we see.
It must be, considering the relative snobbery within the biomedical science field, and how the humans generally tend to look down their noses at some of those in the non-human areas. It must be a pleasant surprise or a pleasant change to be on the other side of that equation occasionally.
Well, we're never really on the opposite side of that, because we're kind of like the poor relations from across the track come driving up in the Beverly Hillbillies kind of truck with Granny on the back in a chair. But they at least listen to us more than they used to, and they see, they're like, we see your value now, but you're getting our carpets dirty because you're dripping dirt and mud everywhere. But that's okay. I mean, I've reached the point in my career where that snobbery doesn't really bother me very much anymore. I mean, it did at one point, but at this point, you just kind of got to laugh and enjoy it, because that's one of the great things of the world of animal science and ruminant nutrition especially, is it's such a small world and it's a family, and everybody kind of talks to each other and listens. And I could call some of my competitors right now and they'd send me samples and they'd help me out, and vice versa. And it's really one of those special things about working in agriculture that is not discussed enough, I don't think, because I've got friends in the biomedical research and they will all cut each other's throats in a heartbeat. Me, I can go see my friends, whether you're talking people outside the US or inside, and they'll give me free run in their lab and vice versa. And it's one of those things that we just don't talk about enough.
That is really, really excellent to hear. And I think, with the current challenges that society's facing in terms of feeding everyone and maintaining the sustainability of our wild systems and farming systems, there's kind of bigger fish to fry, I would say. But you mentioned earlier the concept of what is, or the question of what is a good microbiome. And can we dig into that a bit deeper? Because I imagine it's not about species X, Y, and Z together, but are there some higher level things in terms of how it collaborates or communicates, or something beyond my understanding? But just if we can unpack that a bit in terms of what are the attributes you'd be looking into that, and then I guess what are the mechanisms to encourage positive versus less positive attributes?
Okay, well, and that's $1 million question. I actually asked that question of my students. I was just writing a student's their graduate defense there, and that's the question that I'm asking them is what is a good microbiome? Because that's going to vary a lot depending on what's your lens that you're using. And I teach them, from a microbial perspective, it's one thing, from the animal, but the human perspective is another. And sometimes those align, sometimes they don't. But to me, if I'm looking in production food animal agriculture, what I want to see is a microbiome or microbial population that does not cause disease symptoms in animals, helps prevent it. So you've got a normal population that minimizes the non-growth energy expenditure. And what I mean by that is where you're not creating inflammation throughout the animal. So you have to have these anti-inflammatory properties of certain microbial species. So we want to have them that will tamp down that inflammatory response. We want to get a good bit of butyrate production, because that's, kind of the holy grail of the moment, where it improves the gut integrity and the tightened junctions between epithelial cells, but it's also anti-inflammatory in nature, and there's some other compounds. So we want to see that, because if the animal's not going through inflammation, they're not wasting their energy on this non-growth energy expenditure. We want it to be, and there's a great paper about 7 years ago now by Shabbat at a University of Illinois, and Brian White and Rod Mackey's group, that was a fantastic paper, and it kind of changed my thinking, because we always thought, evolutionarily speaking, that high microbial diversity was good, just based on, well, that seems like it's a good thing. And what they found was actually the opposite. The most efficient dairy cattle in that study had lower microbial diversity. But they dug a little deeper than just looking at that level. They looked at what were the metabolic end products. And they found that those end products were a little more focused on what the animal could use. And by focused is just more limited. So instead of having a whole bunch of things that animal can't utilize, it was a more narrow spectrum that the animal could use. And the analogy that I use is you're going to build a car. You can build the car in your garage using your tools and your parts over time. It's going to take you a year or more to put a car together, or take me 5 years. But anyway, it's going to take you a long time, versus them putting it together on an assembly line where they can churn out 7 in an hour. What's more efficient? They both get to the same goal. They do it more efficiently. So a microbial population that's a little more focused is like that assembly line worker or assembly line robot now, where it does one job over and over and becomes really specialized. So that microbial population is more specialized at degrading the feedstuffs that animal eats regularly. That's going to improve your efficiency. It also reduces your methane production, which impacts on sustainability. So it's a win-win by doing that. You're more efficient, you reduce your feed expense to the producer, and also you reduce that negative output. So that's what one would look like, is this more focused and efficient for what your goals are. So if you're a dairy producer, you're going to want more milk fat typically, but you're also going to want enough energy to keep high levels of milk production. If I'm a beef cattle producer, I'm going to want one that makes more propionate, but I'm still going to want that butyrate to prevent leaky gut syndrome that's going to cause an increase in inflammation and can potentially allow foodborne pathogens like salmonella to cross from the gut into the tissue. So there's a lot of different things at play as to what a good microbiome really is. And then when you're looking at chickens, say, you got to worry about necrotic enteritis and Clostridium perfringens, or pigs with some of their problems. So there's not going to be just a settled, here's the best population, because then on top of that level of complexity, there is an interaction between the animal's genetics and what host microbial population they have. We do not understand that, but it looks like about 60% of the microbiome population, of that microbial population, is encoded for by host genetics. So there may be a portion of that that we really can't do much about. And it's frustrating and fascinating, because then you're faced with the question of, is there a core microbiome for every species? And people have done a lot of work and they've shown, there's this core microbiome. I'm kind of the outside the box thinker in some ways on this, and people told me I'm full of crap for this. They use another word, but that my argument is that core microbiome, is it really important because it's the same, it's present in all species? Or do we want to look at the ones that vary? Is that your difference? And it's the dog that didn't bark in the night. And the analogy, and you've heard me use this before, is the planes going over, bombers going over during World War II and dropping bombs in Germany, coming back and getting battle damage assessment. They saw all the bombers that had been damaged on the wingtips, so they wanted to up armor that. Somebody said, no, screw those don't matter. Let's look at the spot. And there was one place that was never hit. That's where you up armor, because you're looking at survivor bias. So is this population that we're looking at of core microbiome, is it survivor bias one way or the other? So am I totally wrong? Is that really the most important group, or is it the other ones? My gut tells me it's the other organisms that change, but again, I don't have proof, so I don't know.
So many questions that flow from that. Thank you. That's that is excellent. I think in terms of how we influence it, because I guess whether it's the core microbiome or whether it's, I guess if we take a step back just for a second, and we've got, you mentioned 60% are encoded by host genetics. How much do we understand about the I guess the nurture versus nature side of things? How much are inherited from the mother because they're obviously the close proximity, exceptionally close proximity, so there's obviously direct contact in that regard, and how much of it is actually hard coded into the DNA and that relationship? What do we know in that space?
That's a great question, and I don't think we know as much as we would like to. I've kind of asked that question, and that's where it gets into the geneticists and the statisticians, and they then start speaking languages that I don't totally understand. So my understanding could be completely wrong, but it's hard to separate those things because the dam effect of the mother is encoded in the DNA as well. So you almost have to do these great swap experiments of twins born to embryo transfer cattle and that kind of thing. And no one has done that yet, to my knowledge. I could be totally off base, but I have not seen that study yet. But we know with chickens, because they don't have that maternal contact, that they very quickly pick up a microbial population out of their environment, because they're essentially sterile at birth, or at hatch technically. So they pick up out of their environment, and we know cattle do the same. They're picking up from their dam, from her close contact, from licking her, from her udder. And we know Lilu Kwan did some great work along with Tim McAllister of looking at the skin of the udder and that transfer into the animal on a succession of species in calves. So we know a little bit about that, but the heritability percentage and what's driven by genetics versus environment, I don't know that we're there yet. And I don't know that we know how to ask that question effectively at this point either, because we don't really know what to look for.
Thank you. That makes sense. In terms of that nature or the natural, the skin of the udder, etc., how is that? Do we know much about the environmental connections there? Obviously, if a cow is outside, they're sitting down on the grass, and that's interacting with the soil, and that's interesting with the plant biota, etc. Do we know if there's correlations between, obviously soil is related to the rock types, etc., that the soil is made of and the species of plants that grow there. So there's a complex microbiome there in that space. Do we know about how that affects the animal microbiome?
There's people that are looking at that connection between the soil microbiome and crop and animal. We've done a little looking at things like irrigation water, the cleanliness of it, what its microbiome is versus what goes in the animal. And our results were pretty bad, honestly. They didn't make a lot of sense. There wasn't a connection that we saw from that irrigation water. But really, by the time an animal reaches maturity, or at least not maturity in physiologic sense, but in the rumen sense, that's a pretty complex population in
So
the
you're talking 3 months, give or take?
Yeah, at about 3 months, yeah, they pretty much they're functionally a ruminant by that point. So up until then, it's a lot of their environmental impacts, and things we don't think about are like migratory birds that come in and defecate in the feed bunks, and the animals can eat that and they can pick up these pathogens. So if an animal encounters a pathogen, if a calf does, before that rumen is totally put together, that has an opportunity to survive and potentially even thrive in there. Whereas when that rumen population is relatively established and kind of reaches its own equilibrium, then a pathogen is going to have a harder time penetrating that population. It's not impossible, but it's a little more difficult.
So there could be a seasonal variation as well.
Oh, definitely.
So a spring calf could end up with a different balance within their microbiome, say, than an autumn or a winter or summer calf.
Yes, they can, because you've got sunlight, you've got different species that you encounter, different feedstuffs that you're feeding at those times. So yeah, you can wind up with very different microbiomes in these animals.
What do we know about how all the species and bugs within those microbiomes, I guess, interact, and by that I mean collaborate, and the conflicts between them and how they communicate? And does that lead to potential mechanisms where we can intervene to tweak the fermentation in ways that we would find desirable for our purposes?
Yeah, that's a lot of the work that we, along with a lot of companies, are doing, is how do you change this microbial population? The big driver, as always, is diet, because diet is like changing the weather. If you change to a higher grain ration, or you go to steam flake corn from whole corn, you're going to change the availability of starch. That starch is going to increase your lactic acid bacteria, which are going to make more lactate. It's going to drive the pH down. You'll get a little more propionate, but that's going to drive a drop in your acetate and your fiber degradation. So you change that microbial population, just something as simple as changing the form of your grain. But because that'll drop your pH, that's going to be a global change on your microbial population. So diet's one of your big drivers, but you can also add in microbial populations. We do that frequently with what are called eubiotics now, what we always used to think of as that traditional live probiotic. But typically, those need to be added every day to maintain their share of the population. So you've got to add them in continuously, so they're effectively a constant transient passing through. We can also use prebiotics and what are now being called postbiotics, again, another form of what we used to call probiotics, that those can actually stimulate certain members of the population, give them a competitive advantage. Things like malate or fumarate that cause bacterial species in the rumen, like Selenomonas ruminantium or Megasphaera elsdenii, to increase their uptake of lactate. So all of a sudden, instead of this lactic acid, they're converting it to propionate, which is good for the animal. So you can you provide them that competitive advantage. They take up a larger portion, and you change the weather again, so to speak. And there's others that are things like yeast products that are cellular wreckage, but some of that will change the microbial population. Some of it is anti-inflammatory for the animal. And there was a great study that came out again about 6 years ago where they looked at dairy cows that they provided a probiotic to. And they looked at dairy cow gene expression by the host animal, not by the microbes, just at the host animal. And they found just by feeding a probiotic compound, and I don't remember what it was, they had 11,000 host genes changed in their regulatory capacity. Like 9,000 went up, 2,000 went down. And most of the ones that changed were involved in inflammatory response, toll-like growth receptors, and growth hormone. So there's some really profound communication of some form between the microbe and the host. The microbes themselves exchange so many metabolic intermediates of like B vitamins, because we don't have to supplement cattle with B vitamins because the microbes take care of that. But they're not doing it for the animal. They don't know the animal's there. They're providing them to other members of the microbial population who will, say, break down forage that B vitamin producer needs something from. So they are doing these kind of quasi beneficial reactions with each other, but they don't know about each other. They don't care. They just do X, Y happens, and they get what they need. So they're not being altruistic in any sense, but they work together to degrade that feed stuff unintentionally, but it benefits the animal as well. So there's a lot of crosstalk and beneficial interactions. You also have antimicrobial production in the rumen and the gut throughout. Bacteriocins, other antimicrobial peptides, even antibiotics being produced in there that are medically important antibiotics being produced by some microbes potentially, where are some that are related. So that changes that microbial population. It's a really rough neighborhood. In spite of all the hold hands, sing kumbaya, let's work together, they're still trying to murder each other as well. So the microbes are fiercely competitive in a lot of different ways. They use a lot of different strategies, and cooperation is one, attempted murder is another. They do all kinds of things to one another. So it's not a very nice environment in one sense, but in another, it's very cooperative, because you've got, in the rumen, you've got at least 2,000 species. I think it's closer to 20,000 that are operating at the same time. And they all have different niches they occupy, different strengths and weaknesses. So, even if you're just looking, say, at lactic acid bacteria, the ones that ferment starch, depending on what's going on, the relative proportions will change. Your total lactic acid bacteria may stay the same, but certain members will win, certain members will lose out of that same niche. But there's a lot of duplication across. So it makes this microbial population very plastic, that it can change a lot without causing an impact on the animal, or it may cause an impact on the animal, depends on the change.
And I imagine that's both an opportunity and a potential issue or a challenge. And one of the things, from my understanding, a bacteria or a microbe isn't like us. It doesn't eat something and then digest it inside it. It digests it extracellularly outside it. So it secretes an enzyme to a certain extent.
Well,
And is that
it
how?
depends. It depends on the organism. Some of them do secrete their enzymes outside and they release it. Some keep it right up against their body, against their cell membrane. Others, they will bring, like, say, a peptide or a small protein or a small group of sugars inside the cell and metabolize it in there. Most of the true fermentation happens inside the cell because they don't want to lose those intermediates. But that breakdown of the larger compound of going from forage from a blade of grass to something the microbes can actually take in happens outside the cell. So it's a mixture of both. You've got a breakdown process, but that's not actually the microbes utilizing it at that point for the most part. So everything goes through a very stepwise breakdown from big to medium to small to tiny. And at the tiny point, it can be taken up by those microbes.
And I guess there's hundreds of thousands or tens of thousands of steps and microbes.
There can be, yes. I mean, there's at least 7 or 8 layers, trophic layers, when you're looking at the breakdown of forage. So it's broken down into a lot of different things. There's interactions and cross feeding. And ultimately, like for forage degradation, methane production helps speed that process up. So methane production is actually beneficial to forage degradation. They're really inextricably intertwined, but that's something that we look at and we're like, well, that's over most of our heads. But
can you explain that
sure.
A bit more?
As you're breaking down forage, the process that it goes through tends to produce a lot of a reducing equivalent. And I'll get a little biochemical here. NADH tends to accumulate because the rumen is very anaerobic and very reduced. So getting rid of NADH is necessary for the cell to be able to metabolize that forage, for the microbe to be able to metabolize it. So it's got to take the NADH back to NAD. So it's got to get rid of that hydrogen. Now, the rumen has a very low partial pressure of hydrogen gas in it normally, and it's kept really low by the methanogens. And they keep it so low that if you're growing rumen bugs, like Fibrobacter succinogenies in pure culture by itself, it produces a lot of ethanol. That's where it's dumping the NADH, where it's dumping the hydrogen. But if you grow it with a methanogen, it doesn't produce ethanol, it produces more acetate. So it shifts, instead of dumping the hydrogen onto ethanol, it dumps it onto the methanogen. It makes methane, and it allows that bacterium, Fibrobacter, to make more acetate instead of wasting carbon as ethanol, makes more acetate. So it gets a third more energy out of each glucose simply by having a methanogen around. So the methanogens effectively help pull those biochemical reactions forward. They make them more energetically favorable across the board. So this anaerobic environment is really helped by methanogenesis. So it really does, and that's why if we come up with something that wipes out all methane production, we're good, right? That's going to cause a major problem. We'll have to find some other reducing equivalent sink that the microbes can use. And this is why when you look at pictures of protozoa, you'll notice there's smaller microorganisms all over them. And those are methanogens living on the protozoa, living on the fungi, living with the bacteria as well. It's a very symbiotic relationship because they are really just sucking that hydrogen off to make methane, but it helps that organism. It's kind of like a remora of swimming with sharks.
Wow. That is,
it's a lot more complicated than you think.
No, that's being explained much more clearly than I've ever heard it before. So that is excellent. One thing that has come up in multiple conversation is, having acetogens out competing the methanogens, which happens in some species like kangaroos and ostriches and some termites, etc. It's very simple for us to say, cool, let's just support the acetogens rather than the methanogens, as though they're two different football teams. But it's more complex than that. How do you see that relationship?
Well, the acetogens don't grow as fast as the methanogens. So that means they don't metabolize as quickly, unless there's something that's changed that I haven't seen. So, I mean, this is, again, my knowledge, and I'm always somewhat limited. But the acetogens just don't grow as fast, so they don't keep that partial pressure of the rumen as low. Plus, they're struggling with their doubling time to be able to maintain their population in the rumen, because remember, there's a constant flow in the rumen. It's like living in a barrel where you got a hose coming in and the hose going out. You've got a constant flow. So if you can't double your population, at least at that flow rate, you lose. You get washed out of the rumen eventually. So the acetogens, it's a great idea, but we've got to do more with them where it speeds their growth time up. And I know people are looking at that, but I don't know how well that's going. I don't know if that's something that really can be changed.
Are you excited by the potential of synthetic bio or that sort of stuff being able to come in to that space and tip the balance?
It depends on what you mean.
Or another tool.
Well, it depends on what you mean by synthetic bio.
I mean, emerging technologies that weren't around 10, 15 years ago, whether that's tweaking the genome of an acetogen, whether it's you inserting the acetogenic part into a more rapidly growing species, whether it's just understanding what cofactors are needed to make the acetogens more competitive or understand the biology at a much more molecular level. I mean, that
okay, well,
space.
Well, I think using a genetically modified organism is always or for the foreseeable future, is going to be kind of a no-fly zone, because people will freak out about that, whether it's justified or not. It's above my pay grade. But I know that's going to be something that creates that, turning it loose in the wild. Doing it in the lab, sure, we can do that, and people are, and it works great from what I understand. But I don't know how much they're actually going to allow that to go on into the real world. But understanding what their role is and what their metabolism is, and then exploring other environments, is going to be important, because we keep finding these extremophiles in weird environments that we may be able to use them, or we may be able to use specific enzymes and use enzyme preparations rather than the live organisms, or use cellular wreckage from those organisms, where we're not adding a live GMO or a live set of genes that can be transmitted, that we're just adding this ingredient that is an inert, non-living compound that won't transfer resistance or any horrible thing. That has some real possibilities to do that, to stimulate these populations. And things like the 3-NOP that is being looked at to reduce methane production. It's not going to wipe it out, but it slows it down dramatically. And things like Andre Wright had done some vaccination studies against methanogens. You can make these changes, but we've got to find something. And this is where having organisms that produce more propionate, because propionate is a very reduced end product as well, that we can shift some of that production to propionate. And that's a biologically relevant end product instead of methane. It's important to the animal. So with that case, if we can shift that way or towards acetate, that's something the animal can use, and that helps improve our efficiency and ultimately that energy capture from the diet.
And from my understanding, I think that the a final solution is quite likely not going to be just one of those things. There could be the 3-NOP or some similar thing changing the competitiveness of the methanogens, which then gives more opportunity for other bugs to come in there because you've just changed. You've given them a bit of a boost.
Yeah, there is no silver bullet. I wish there was. I mean, for years we thought Monensin, and I love Monensin. I think it's a great molecule. It's something that changes that microbial fermentation so well without the impacts on antimicrobial resistance. I mean, it's a cool molecule. That was what I worked on for my master's and PhD. So, I mean, yeah, I think the world of it. And that was the closest I think we'll see to a silver bullet. But even it had limitations. We're going to have to use multiple hurdles, multiple approaches simultaneously. And it's going to vary, and it may have to eventually be almost tailored to the farm or at least the phase of production, like, you know, grower cattle versus feedlot cattle versus dairy cows versus calf versus pasture dams. You know, you may have, 6 different treatments that you use on a farm.
And to think a little more globally, in terms of livestock production, especially ruminant production, be that dairy or beef, within the broader community and the media, it's the methane conversation that is by far consuming the most column inches and the most airtime.
Sure.
And I don't think there's anyone who disagrees that that's a big challenge and something we have to address. How do you see that balance? And are there other things within the sustainability conversation that do need to, or you think should have a little more airtime?
One of the things really, I mean, that's a great question. One of the things I like to look at still is ammonia production, nitrogen runoff, because that still is an environmental pollutant. It's very impactful at a local and semi-global because you're pouring it down, say, the Mississippi River, and you get algal blooms in the Gulf of Mexico that can be very potent. So that nitrogen flow is a big deal, and it's one that, it's like methane. It comes and goes in importance as well. But, clearly the methane is a big one. I'm still, I've done a lot of work in food safety. That's one of the big issues as well, is how do we make sure we still have that we're ensuring food safety while we're doing this. But, those are the big ones that I run, where my heart lays with those. But again, since I grew up doing Monensin stuff, so to speak, I'm always going to look at that impact on both methane and nitrogen production or nitrogen release and products like that. And, we don't even really know what happens if you do 3-NOP methane at the same time. We don't know what's going on to these populations and how it affects the dynamics of that hydrogen flow.
But you meant 3-NOP and Monensin rather than
right.
Yeah.
Oh, I'm sorry. Yes, I'm sorry.
Yep. No, just wanted to clarify that.
Yeah, you're absolutely right.
When we think about the production systems here in the United States and the number of cows on feed in terms of beef cattle, the intensity and the high touch of the dairy industry, there is a lot of touch points, daily touch points, where we can add additives, where we can change the diet to actually tweak what's going on in the rumen. Whereas when you think more broadly across the globe, in Australia where I grew up, or India has more cattle than any other country in the world, and they're virtually all smallholders, and there's the touch points are much more complex in terms of getting access to make changes. How do you see, I guess, the solutions that are being developed being applicable to those hundreds of millions of cattle that are more in grazing environments?
Well, and that's one of the issues that we have. A lot of our cattle here in Georgia are pasture grazed cattle. They have calves. You handle the calves 3 or 4 times in their life, and then they go down the road. Yeah, that's difficult. I mean, there's not a silver bullet. We're going to have to, the low hanging fruit are going to be the ones that we can hit first. But, your dams out on pasture where they're grazing their entire lives, their entire productive lives, that's going to be really hard to do much with, because a lot of these farmers just simply don't feed them. Or if you look out west, where you'll have 10,000 acres and 1,000 cattle, that's going to be really hard to do anything to, unless you can do some kind of genetics or vaccination. And the vaccination against methanogens has potential, but it's not there at this point. I think there's opportunities long term, but it's going to take time for us to sort all that out. So there are potentials like that, and including things like liquid feed supplements or lick tanks, where you can get some of the 3-NOP or metanensin or some other compound into them in a slower release form, has potential as well. But there's going to simply be some animals that we just can't reach effectively.
Where, if I had the blank check, or if to give you, where do you see more attention needs to be or should be directed? And that could be investment, it could be more researchers, just more capacity to do the research, it could be collaboration, it doesn't have to be just a financial investment in a very broad sense of the word. Where do you see the priorities or the needs in that space?
So my Ferrari fund wouldn't be that then. Okay. Well, some of the work that we're doing with one of my colleagues that just left here and went to Florida, is where we've been looking at the genetics of beef cattle and selecting like high marbling lines, low marbling lines, and high feed efficiency, or high average daily gain, low average daily gain. So you've got 4 groupings there, and looking at their microbiome and which microbes are correlated both in the rumen and the hindgut of these cattle, and trying to understand who's related to these different factors, because you've got impact on yield grade based on total back fat, but marbling impacts quality, which is your payday.
Yeah.
So looking at that for beef, and then something similar, milk fat versus total milk in our Holsteins, and looking at that genetic correlation. Are there, since there's 60% of the microbiome is correlated from this microbial population by what the dam or the sire have, do we have lines that are simply going to be better microbiomically than others? Do we need to start focusing on them? So can we come up and look at an animal, pull it up in the chute, do a fecal sample and or a ruminal sample, and say, this has got a good microbiome, we don't need to do anything. This one's got a bad one, let's give them product X. This one's got a good one in the rumen, it's got a bad one in the feces, let's give them product Y. That I think that's one of the places where it's almost tailored medicine for these animals. But that's going to require an investment in time, contact, working with the animals specifically. I think really that's going to be one of our big limitations, is just the workforce that's able to work with these large, dangerous animals. Because, most of us that have worked with cattle along the way have gotten, broken bones, broken ribs, all that. Not everybody on the planet wants that. Some people tend to be discouraged by getting a broken arm. Some of us aren't very smart and we keep doing it, but that's life. So that's going to be an area that I really think we need to get some better connection with people and their food supply, because we are becoming so siloed that I always thought it was a joke, but people really, kids really think their food supply comes from the grocery store, that there's nothing supporting that. And like I said, I didn't think that was real. Now I see it's real, and I'm kind of horrified by that. So that's the thought in the back of my head that really keeps me awake some nights, is how do we fix this kind of problem?
So that's on the human side of it, and us
Yeah,
understanding, I guess, more about our world than the four corners that we live in, or the
exactly.
neighborhood we live in, and to bring, I guess it's part of its empathy, part of its an understanding of the challenges, those sorts of things.
Mhm. Yeah, I mean, we've done such a great job. And what I tell students is we gave farmers a charge, 300 years ago when the Malthusian theory first came out that, there's not going to be enough food. Farmers, produce as many calories and as much protein as you can, as cheaply as you can. Go. And our farmers have done that. I mean, they're so successful that they've harmed themselves because there's that disconnect. And that cheap food supply enables our standard of living, but people don't realize that they're almost a victim of their own success.
Yeah, one of the biggest hurdles to overcome is the previous success to be able to build
Exactly.
the population up to 8 or 8.5 billion or whatever it happens to be.
Yeah, exactly.
How do you see, students and their attitudes and their concerns, I guess, changing over time, and the opportunities of these young people that are coming through, and their energies and passions, and how that can be harnessed to drive these positive changes you're talking about?
Well, the interesting part is, people try to break down these generational differences. And I hear a lot from, I'm Gen X, so I hear a lot of people in Gen X of millennials and Gen Z suck because whatever. And, I've gotten to see those kids, and they're just as smart, just as hardworking and dedicated as any other generation. It just looks different. Because for our generation, we would talk to people going to class. They don't, they're texting with somebody else that's not there. So it's the same thing, but it looks different. So we tend to, the older generations tend to look down on the younger ones, but these kids want to know. And I've got a class for first years, and I teach them a lot of random things, but a lot of it is about, where their food comes from, where their food supply comes from. And the number of these students from big cities that are like, this is really cool. And they want to get into it. They want to know more because there's such a dearth of information on where food comes from. And there's so much just propaganda that these students see. And they're better than we think at telling I'll just go and say it, telling bullshit from truth. And you can edit that out as you need to. If you don't, that's fine too. But they're really good at detecting bullshit at this point. And they can spot that better than what we think. And they respond so well to that earnest reality and that truth telling that, that's our job. And when they can see this is the reality, they respond so well and they get engaged in it because millennials and Gen Z want to solve problems. That's their goal. They're kind of, for want of a better term, and it's always dangerous to say things about a generation, but they're kind of crusaders. They want to make the world a better place. And at this stage where they've got the enthusiasm and the energy and the belief they can do it, and there's really no greater force in the world. And I mean, I know that's idealistic, and I
No,
understand
that's right. And I guess when it comes to using technology to leverage and amplify their messages, they're probably a hell of a lot better than you or I.
Oh yeah, they're fantastic.
At that. That's really, really cool. Is there anything else that we haven't covered that you think should be added or you would like to say?
No, I think that's pretty much covers a little bit of everything.
Awesome. Well, It's been an absolute pleasure chatting to you. So thank you very, very much, Todd.
Thank you, Ash. I appreciate it. Thanks for listening.
You've been listening to The Ash Cloud with me, Ash Sweeting, in conversation with Todd Callaway, recorded in California in April 2023.